Hello everyone. I'm Changhyo Ahn, who presenter about this journal. The title of this journal is Performance and Bacterial Communities of Successive Alkalinity-Producing Systems in Passive Treatment Processes Treating Mine Drainages Differing in Acidity and Metal Levels. The order of presentation consists Introduction, Material and Methods, Results and Discussion. Introduction. Mining activities in coal mines expose sulfide minerals to the atmosphere and water. In this process, mine drainages harboring high levels of acidity, iron, and sulfate are created and have become a representative mining hazard in aquatic environments. First, we need note a meaning of SAPSes. SAPSes is successive alkalinity producing systems, and it is key unit process in a passive treatment process of acid mine drainage, performing acidity neutralization, sulfate reduction and subsequent metal precipitation. Initially, SAPS was installed to enhance metal hydroxide precipitation by increasing pH and providing alkalinity. However, biological sulfide production and metal sulfide precipitations were observed, which were found to be more efficient metal removal mechanisms. SRB utilizes a variety of organic carbon sources. Then various substrates were tested for increasing SRB activities. There have been many indirect investigations of SRB activities and communities in AMD passive treatment processes. However, more microbiological research is necessary to understand SAPSes better in order to make them more efficient. Two passive treatment processes with significant differences in acidity levels and metal concentrations were selected to deepen our understanding of SAPSes by characterizing bacteria communities in very different SAPS conditions. Because the two processes are quite different in their performance, it was expected that unique SAPS bacterial communities could be characterized. Follow this process. This study will be a basis for provides valuable information on bacterial community structures in the AMD treatment research field. Material and methods. The AMD passive treatment processes in Gapjeong, GJ, and Seokbong, SB, were constructed to treat AMD from abandoned coal mines of Mungkyong City in South Korea. The investigated site exhibits typical mountainous climate, with 20.0 degrees C average temperature, the 23rd of August 28, 6 degrees C, 308.7 millimeters of rainfall, and 79.4% humidity in July. A typical SAPS consists of a 1m high limestone layer, Caco3, a 0.5m high organic material layer, and a 1 to 1.5m of overlaying water. Analysis of pyrosequencing data CD-HID was used for massive clustering of metagenomic sequences. Richness and diversity indices were calculated using MOTHA 1.280. Analysis of variance, ANOVA, was performed using SPSS 18. The multidimensional fast unifrac distance matrix was converted into two vectors using principal coordinate analysis. 6,169 sequence reads were randomly selected from the bacterial sequence pool of SAPS SB 5 times using SEAL Community 3.0 for accurate comparison of the bacterial community structures. Operational taxonomic units, O2s, were defined at the 3% divergence threshold or 97% similarity. Result and discussion. AMD pH values were very different in two sites, where GJ had pH 6.73 and SB had pH 3.16. Influent levels of Fe2+, Al3+, Mn2+, sulfate, acidity, and electric conductivity were much higher in SB than GJ. In influence of GJ and SB, sulfate concentrations were much higher than total iron concentrations. Considering that iron and sulfate are mostly produced from pyrite, a considerable portion of the initial iron might be precipitated as AMD flows through channels to the passive treatment processes. In terms of alkalinity production and metal removal, the performance of SAPS SB was in the recommended performance ranges or exceeded them, while those of SAPS GJ were lower. Metal cations in AMD react with sulfide and precipitate as solid metal sulfides. These metal sulfides typically have a very low solubility in water under anaerobic conditions. The blackish color of the sediment from GJ demonstrated the pyrite precipitation. Possibly due to the active SRB activity, the reddish sediment in SB might indicate the absence of the pyrite precipitation. However, the physicochemical analysis demonstrated that SB performed metal removal and alkalinity production very well. This indicates that diagnosis of SAP's performance merely based on sediment color can be misleading. They proceed pyrosequencing analysis to SAP's bacterial communities. Pyrosequencing is a method of DNA sequencing based on the sequencing by synthesis principle, in which the sequencing is performed by detecting the nucleotide incorporated by a DNA polymerase. Pyrosequencing relies on light detection based on a chain reaction when pyrophosphate is released. Hence, the name pyrosequencing. High-quality sequences longer than 300 BP were recovered for more accurate taxonomic identification, and these were 2-3 times longer than the minimum lengths in previous studies. This figure shows that taxonomic compositions at the 6 taxonomic ranks demonstrated that bacterial communities of SAP's sediments were distinctly different between GJ and SB. Chloroflexi, firmicutes, and proteobacteria were the 3 most dominant phyla in GJ. Firmicutes was the most dominant phylum, followed by acidobacteria and proteobacteria in SB. In the class level, Anaerolini and bacilli in SB were the most dominant class in GJ and SB. Cranarchaeota in the Archaea domain. Among them, Delta Proteobacteria and Clostridia are the 2 most predominant classes affiliated with the SRB isolates, which were also found in the sapses of GJ and SB. In the sediments of the tested sapses, Clostridia and Delta Proteobacteria were the 5th and the 6th largest classes in GJ, and Clostridia was the 3rd largest class in SB. Most of the bacterial sequences were found to be uncultured in the species level. SAP's bacterial communities of GJ and SB had 190 O2s in common, which occupied 9.7 and 11.9% of total O2s identified in GJ and SB samples. And low portions of shared O2 suggested that bacterial communities were not similar in SAPS GJ and SAPS SB. Bacilli and Clostridia were the major classes in the shared O2s, in which Bacillus, Licinibacillus, and Uribacillus were the major shared genera. Diverse and complex bacterial compositions were found in our study. Because SRB cannot utilize complex organic carbon source directly, synergism of various trophic groups such as acidogens, methanogens, and sulfate reducers is essential to provide short-chain organic carbon compounds for SRB. And diverse bacterial trophic groups are known to be involved in sulfate-reducing bioreactors fed with complex carbon compounds. A culture-based analysis revealed that non-sulfate-reducing bacteria were dominant in compost-based sulfate-reducing bioreactors. However, in a methanol-fed bioreactor treating AMD, the bacterial community was dominated by sulfate reducers, mostly affiliated to Delta Proteobacteria. Because methanol is a simple organic carbon source, a complex trophic group might not be necessary to oxidize complex carbons. The sediment bacterial community of SAPS GJ was more diverse and more evenly distributed than that of SAPS SB. CDHIT-based rarefaction curves showed that the richness of the sediment bacterial community of GJ was higher than that of SB. And figure 4c shows that more than 99% bacterial O2s had less than 1% abundance in the total bacterial sequences. As you can see it in table, richness estimates of GJ were higher than those of SB. However, an ACE value of GJ was lower than that of SB at the maximum sequence reads, suggesting that diversity comparison at different sequence reads biases the microbial diversity analysis. And diversity estimates proportional to richness and evenness were higher in GJ and SB as well. Simpson were 7.120, 6.879. and 350 in GJ and 6.344, 6.116, and 117 in SB, respectively. CD hit rank abundance curves show that more than 99% bacterial O2s had less than 1% abundance in the total bacterial sequences. Arrangements of unit processes differed in the two treatment processes. The SAPS in SB was located before anoxic pond because the high acidity of AMD in SB should be neutralized first. Anoxic pond in GJ was located before SAPS GJ because pH of OPGJ was still high. The blackish sediment color and cleaner effluent in the SAPS GJ indicated that the SAPS GJ performed better than the SAPS SB. However, the area-based treatment rates showed that the SAPS SB had better treatability than the SAPS GJ. Because AMD in GJ harbors much lower acidity and metal loading, total treated amounts of metals and acidity were also much less in the SAPS GJ, where metals and acidity were almost completely treated. Notable differences were observed in bacterial community compositions of the two SAPSes in GJ and SB, with drastic differences in acidity, metal concentrations, and treatability. Bacillus was the predominant class in both SAPSes, and it is not affiliated with the currently known SRB. The molecular evidence of Bacillus in our study demonstrated that the isolation and understanding of bacilli-affiliated bacteria in SAPSes are expected to promote the understanding of SAPSes. A high portion of uncultured sequences will necessitate exploring the physiology of new isolates in SAPSes to understand their microbial ecology better. The sediment bacterial community of the SAPS SB was simpler and less rich than that of the SAPS GJ. Bacterial communities in controlled bioreactors for specific purposes tend to be simpler. So, bacterial community structures indirectly indicate that the SAPS SB performs well as designed, as indicated in its physicochemical performance.